Hello everyone, welcome to 10-Minute Physiology. In today's video, we're going to talk about how thyroid hormone is synthesized. So with that, let's give it a go. So I'd like to begin by first talking about how the thyroid gland looks in the cellular level. So this right here is a diagram showing you the cellular structure of the thyroid gland. And what we see here are a few important structures. The first one is going to be the follicle. So the follicle is going to be the circular structures that are basically made up of epithelial cells called follicular cells. So follicular cells are cuboidal epithelial cells that make up the epithelial lining of the follicle. Now these epithelial cells, the follicular cells, divide up two fluid compartments. The first compartment is going to be the colloid, which is the lumen inside the follicle. And then the second compartment is going to be the interstitial fluid, which is outside the follicle. Now the interstitial fluid is going to surround a bunch of different structures. The interstitial fluid surrounds the follicles, and it also surrounds the parafollicular cells or C cells. In addition, the interstitial fluid is also going to be surrounding capillaries, which bring blood into the thyroid gland, allowing thyroid hormone to enter into circulation. And we also have the interstitial fluid, which basically bathes all of these different structures. So it bathes the capillaries, the parafollicular cells, and the follicles. So now that we have an understanding of the cellular structure of the thyroid gland, let's take a look and see how thyroid hormone is synthesized by the thyroid gland. So this is the diagram that we're going to be using today. So this green rectangle is going to be the follicular cell. And this follicular cell is dividing up two compartments. The first is the colloid, which is the space inside the follicle. And then the second is going to be the interstitial fluid, which is outside the follicle. So now, since we know what this diagram is talking about, let's now get into how thyroid hormone is synthesized by the thyroid gland. So the first step in synthesizing thyroid hormone is to get iodide into the colloid. So in order to get iodide into the colloid, on the basolateral membrane of the follicular cell, we have the sodium iodide symporter. So the sodium iodide symporter co-transports two sodiums and one iodide from the interstitial fluid into the cytosol of the follicular cell. So once the iodide is in the cytosol, the iodide has to be transported across the apical membrane into the colloid. And in order to do that, the follicular cell uses pendrin. So pendrin is an anion exchanger, and what it does is it exchanges one iodide, which moves the iodide into the colloid, for another anion. So pendrin allows the iodide to move into the colloid. And once iodide is in the colloid, iodide can be processed more and then used to form thyroid hormone. So also in the cell, we have thyroglobulin. So thyroglobulin is the precursor protein that is used to make T3 and T4, which are the thyroid hormones. So thyroglobulin is going to be synthesized by the cell's secretory pathway, and thyroglobulin is going to be packaged in vesicles. These vesicles will fuse with the apical membrane, which will allow thyroglobulin to enter into the colloid. So this right here is a short representation of thyroglobulin. The real thyroglobulin protein is going to be extremely massive. It's going to be around 600 kilodaltons, and it has around 100 tyrosine residues on it. And only around 20 of these tyrosine residues are going to be used in order to form the thyroid hormone. So there's tons of residues on this protein, so it's a massive protein. So the next step in this is that we need two enzymes, and these enzymes are going to be TPO and DUOX2. So TPO and DUOX2 are going to be very important for the next step in synthesizing thyroid hormone. So right now we basically have our precursors or our ingredients for thyroid hormone, which is thyroglobulin and iodide. However, in order to allow iodide to react with the residues on thyroglobulin, we need to oxidize iodide into iodine. And the way that this is done is through a two-step process. So the first step is that DUOX2 catalyzes the conversion of peroxide into hydrogen peroxide. The hydrogen peroxide is then used by TPO to oxidize iodide into iodine. So iodine can then be used in order to iodinate the specific residues on the thyroglobulin chain. So the residues on the thyroglobulin chain and iodine react together to form iodinated tyrosine residues, which we see right here. So what we see with these tyrosine residues is that they're iodinated by 2 iodines on each, and each of these residues is going to be called DIT. The reason why it's called DIT is because there's 2 iodines on these residues. Now note that you can also have MIT form. So MIT is only when you have 1 iodine on a tyrosine residue. So these right here are just showing DITs. So after these DITs are formed, the DITs can undergo a conjugation reaction that is going to be catalyzed by TPO. And this conjugation reaction basically connects 2 of these residues together. So what we see here is that these 2 residues are now connected together, as we see right here. So this residue is going to be called T4. So this T4 that we see right here is still connected to the thyroglobulin chain. And basically what's going to happen is that T3 and T4 are going to be stored in the colloid while they remain on thyroglobulin. So this right here is a summary of the steps that we just talked about. So as a review, iodide is taken from the GI tract, enters the bloodstream, and reaches the thyroid gland. Iodide is taken up by the follicular cells through the action of the sodium iodide symporter on the basolateral membrane. Iodide is transported out of the follicular cell into the colloid by pendrin, an anion exchanger, while thyroglobulin is released into the colloid by the secretory pathway along with TPO. TPO uses hydrogen peroxide produced by DuOx2 to oxidize iodide to iodine. TPO also catalyzes the conjugation of specific tyrosine residues that were iodinated. And then thyroid hormone still on thyroglobulin is going to be stored in the colloid lumen. So now what we're going to do is we're going to look at the next set of steps that the follicular cell is going to use in order to secrete thyroid hormone into the bloodstream. So the first step is that we have to remove the thyroglobulin that is stored in the colloid and bring it into the follicular cell. And the way that the follicular cell is going to be doing this is through fluid phase endocytosis. So basically, through fluid phase endocytosis, thyroglobulin is taken up and it's packaged in these vesicles. These vesicles then fuse with the lysosome, and the lysosome is then going to hydrolyze thyroglobulin into MIT, DIT, T3, and T4. So T3 and T4 are then released into the cytosol, and T3 and T4 can then be transported through facilitated diffusion by MCT8 into the interstitial fluid, and then T3 and T4 can enter into the circulation and be circulated throughout the body. Now, it's important to realize that the follicular cells are going to release varying amounts of T3 and T4. Around 10% of thyroid hormone that is released by follicular cells is going to be T3. Around 90% of it is going to be T4. And there's going to be minimal amounts of reverse T3 being released by these follicular cells. So the last thing I want to talk about is the effects of TSH on all of the steps that we talked about today. So the first thing that TSH can do is it can increase the activity of the sodium iodide symporter. Therefore, TSH can increase the amount of iodide trapping by the thyroid gland. In addition, TSH can stimulate iodination of the tyrosine residues on the thyroglobulin chain. TSH can also stimulate the conjugation reactions that are catalyzed by TPO. So it can stimulate the conjugation reactions between different tyrosine residues, therefore allowing more thyroid hormone to be formed. TSH can also stimulate fluid phase endocytosis, which allows more thyroglobulin to be taken up from the colloid. TSH can also increase proteolysis in the lysosome, therefore allowing more T3 and T4 to be hydrolyzed off of thyroglobulin. Also, TSH can also increase the transport of T3 and T4 out of the follicular cell into the interstitial fluid, allowing more T3 and T4 to enter into the interstitial fluid and therefore into the circulation. So in summary, thyroglobulin is taken up from the colloid lumen by fluid phase endocytosis. The vesicle then fuses with a lysosome to form a lysoendosome, And inside the lysoendosome, thyroglobulin is hydrolyzed to T3, T4, DIT, and MIT. The vesicle then releases T3 and T4, and they are transported out of the cell by proteins located on the basolateral membrane. And in summary, TSH can affect many of the steps of synthesis. TSH can increase the activity of the sodium iodide symporter. It can increase iodination of thyroglobulin. It can increase the conjugation reactions that can occur between iodinated tyrosines to form T3 and T4. It can increase endocytosis of iodinated thyroglobulin into follicular cells. It can also increase the proteolysis of iodated thyroglobulin. And it can also increase the secretion of T3 and T4 into the circulation by facilitated diffusion. So that's it for this video. I hope this video helped you understand how thyroid hormone is going to be synthesized by the thyroid gland. Thank you for watching, and see you next time.